Hello, everyone, and welcome to the Society for Ecological Restoration's Wednesday webinar series. My name is Alexis Gibson, and I'm the Program Manager for SER. I want to welcome you to today's webinar, Selecting Native Plant Material for Restoration Projects in Different Ecosystems, Successes and Challenges with Karma Buaza. Before we begin, I wanted to give you some information about SER and about how today's webinar will go, in case this is the first time you're joining us. SER is an international organization that advances the science, policy, and practice of ecological restoration to sustain biodiversity, improve resilience in a changing climate, and reestablish an ecologically healthy relationship between nature and culture. One way we accomplish that mission is through our webinar series. To browse upcoming events and information on our many other resources, we invite you to look at our website, www.ser.org. This is part of our Wednesday webinar series, and next week will be the last one of those weekly webinars. In June, we'll be switching back to our more regular monthly webinar series. Today's webinar is being recorded and will be available for future review in our webinar library, which you can also find on our website. So about today, the session will run for around an hour with time for questions at the end. You are all muted, but you can type your questions into the questions box. And once Karma is done with her presentation, I'll be moderating a question and answer session with her where I read your questions and she has a chance to answer them. Our speaker today is Karma Buaza with the Lebanon Reforestation Initiative. Karma is one of our newly elected directors at large, and we're very excited to have her starting in July on our board. She received her Bachelor of Science in Agriculture Engineering and her Master's of Science in Plant Protection from the American University of Beirut in Lebanon. Since 2011, she has worked with the U.S. Forest Service initiative programs in Lebanon, Jordan, Zimbabwe, Morocco, and Rwanda. Through research and collaborative partnerships, she has worked on developing approaches that address the challenges in using native genetic plant material in ecological restoration in diverse geographic locations and climatic conditions. Karma, I'm going to turn it over to you so you can get started now.
Okay. Thank you, Alexis, very much. And hello, everyone. I hope everyone can hear me well and that everyone is safe. Thank you for joining us. And I really cannot wait to start working with SER and such an amazing team in July. So thank you again, Alexis. And so as Alexis was saying, I am very lucky to be able to work with the U.S. Forest Service since 2011 on so many international projects. And today I will just give an overview of some of the projects and results that we worked on, and how we focused on native plant material for restoration in different ecosystems. And if at one point I stop making any sense, please just blame it on the lockdown and, and the whole situation happening around the world. So just as an overview, we're gonna just talk briefly about why using native plant material in ecological restoration. And I'm gonna present 4 study cases from around the world: Jordan, in Guinea, in Morocco, and in Lebanon. And at the end, also we're going to discuss a bit the role of fauna and ecological restoration, which we found it's something that is very important to take into consideration. So we all know that around the world, ecological restoration projects are increasing and the demand in ecological restoration is increasing. So the best way is to ensure that they are successful projects is we have to ensure sustainability and plant material and seedling that is available for all those projects. We try to focus on using native species, and we base a lot of our practices on scientific approach and international standards. So Since 2011, we've been introduced in Lebanon to the target plant concept by Professor Anthony Davis, and I'm sure a lot of you know him very, very well. And the target plant concept was shown to be very successful in a lot of our projects around in different countries. It basically focuses on identifying what are the limiting factors on the outplanting side, framing the problem and the objective of our outplanting, designing seedlings or any plant material that actually can overcome those challenges on the field. We monitor, we record the results, and we try to also, depending on the results, improve seedling quality or practices. And today we're going to focus on two of those objectives or concepts in the target plant concepts, which is the type of plant material that we usually use. Is it seed? Is it seedling? Or are they cuttings? Oh, and the source of the plant material that we use. So to ensure that we have an efficient restoration project and results on the long term, not only successful results this year, we have to ensure that we have successful results and sustainability on the long term. We need to focus on controlling the plant material source and quality to be able to ensure the best efficient restoration practices. So we kind of focus on controlling mainly the quality of the plant material that we use and its origin. I'm going to start with Jordan. The case study in Jordan, we all know that, and Jordan for me now is like a second home. I've been going there since 2015, and I absolutely love this country and the biodiversity that it has. Yes, Jordan has a high flora biodiversity. However, it has less than 1% of forests left, and more than 80%, around 80%, are rangelands, and a lot are degraded rangelands due to urban expansion, agriculture practices, unmanaged agriculture practices such as grazing, and soil erosion. And a lot of the restoration projects were focused on introducing non-native species that threatens the actual biodiversity of Jordan. And we all know that Jordan is one of the poorest countries in water. It has very low precipitation levels, and this is one of the major limiting factors that we face in restoration in Jordan. So when we think about that, we want to use genetic resources that are adapted to this particular limiting factor. So we tend to see which species are drought tolerant, which native species are drought tolerant, and start our restoration process, because it is a long-term process. We start our restoration process with using specific drought tolerant species. And when we started this project in 2016, one of our major challenges were actually using native plant material that we have identified and using them for large production of native seedlings. We're not talking about the quality of seeds for research or in seed banks. We're talking about quality of seeds used in native nurseries for restoration projects. We have found that we have poor storage techniques with very high humidity, poor cleaning of seeds that have that lead to low viability also, and sometimes we have unknown seed sources or species or mixed species, as you can see in the pictures here. In one of the trays, we had two different Atroplex species. So all of this limited the production of quality seedlings. And again, here I want to stress on the larger production of quality seedlings for larger restoration projects, because managing millions of seeds is very hard, and cleaning millions of seeds is very hard. So the project that started in Jordan with the U.S. Forest Service was the SEED project, Sustainable Environment and Economic Development, that now turned into an official NGO, WADI, focused on empowering women and local communities in Jordan, not only in native seedling production and improving their technical capacities and restoration and ecological restoration, not only just for seedling production, but also starting from seed collection to outplanting. So here in this picture, you can see a group of women from different local communities in the northern Badia region in Jordan, in the greenhouse and sowing the seeds that they have worked so hard in collecting, identifying correctly the species, cleaning them, storing them under improved practices, and developing a well-established record-keeping system for seed collection. So here you can see the same group of women working on identifying the species, and also developing ways on counting properly the seeds by weight, cleaning properly the seeds. So it was an intensive few months of training with the local community and with this group of wonderful women. And also we worked a lot with all the local communities to be able, and local experts in Jordan, to identify the best seed sources, to collect the best quality seeds, to be able to manage the seeds and produce quality seedlings. So here just an example of well cleaned, stored seeds to produce high quality seedlings for restoration. And this table is just to summarize. I'm not going to go into the whole details, but these are different species of native species in Jordan between trees and shrub species. And these are the pre-sowing treatment that we developed with the women at the nurseries using very minimal resources available, and that had improved germination rates. Some jumped from 10% to 30%, some jumped to 77% just with a simple treatment, like for the Ceratonia silico. We just dropped them in boiling water to do the scarification for the seeds, and we got a higher percentage of germination. So these are all some treatment that could be done by the local communities, empowering women to be able to do this. So we defined what is our objective. We tried to optimize the nursery practices, produce higher quality seedlings, and then definitely we went to the outplanting trials to be able to measure the success not only at the nursery level, but also on the outplanting trials, and under very harsh conditions. In some places that receive less than 150 milliliter precipitation rainfall. And as you can see here, it's a beautiful picture of an Atroplex seedling that was planted and that received no irrigation during the summer and still survived. So during this project, WADI project was able to successfully restore 856 hectares of land under improved natural management practices, and is just a start. And WADI is continuing to collaborate with all the stakeholders and partners in Jordan to be able just to improve the ecological restoration protocols in Jordan and to make it more sustainable. Another case study is the Guinea case study. This one we focused. Guinea is a country in West Africa, so it's a tropical ecosystem. And one you can see here on the map the Bossou village, and this is the Nimba Mountains, Nimba Forest. This is the corridor between the Bossou area and the Nimba Mountains, where the Bossou area is very important because it is home for the a chimpanzee population that is at risk. And the chimpanzee population is culturally, historically, and environmentally important for Bossou and for the whole country. And they're decreasing at a fast rate due to deforestation and outbreaks of diseases. Um, and they were suddenly genetically isolated from the Nimba Mountains, which is making their movement and mating impossible for reproduction. So the aim here is to actually be able to restore the Bossou corridor. And here you can see the Bossou corridor. And this is the Nimba forest far, far away. So we want to try to restore this corridor to be able to have chimpanzees move from the Bossou to the Nimba area. So we kind of sat down with all the local communities and the local stakeholders, and we thought, what are the criteria to take into consideration for best restoration practices? First, we thought about fast-growing native species. We need to identify which native species can grow fast to decrease competition, because as you can see here, you have a lot of vegetative competition on the land, and we need to actually start with fast-growing species to be able to decrease that competition. We also need to select species that are consumed by chimpanzees. We want those chimpanzees to move from one area to another, so if they don't have that food resource available, they will not be able to move. And our third criteria was to respect biodiversity. So we developed a new system for nursery improvement using all local resources. So the Bossou is a remote area where we have very limited local resources available. So with the help of the local communities and the local stakeholders and the researchers there, the local researchers, we were able to develop a system that uses local resources to improve native seedling production. And here you can see, I love this picture, how they were very innovative in ways to improve their soil, as you can see here, to improve their irrigation management, still using the block weight method and keeping records of it. And here you can see how an improved seedling with improved root systems, and they use different treatments, soil treatments, using different manure treatments also. And they were keeping record of the results to finalize which treatment was best suitable for them. So we had one major challenge again, using seeds or using transplants. We noticed during a germination trial that using the seeds for a lot of species, had very low germination percentages. So as you can see here, they were put on seed beds, and we counted the germination rate for all the species, the date of sowing. We took how many numbers of seeds were sown per seed bed, and we finalized the germination percentage. And most of the species had very low germination percentage, although when we found them in their natural habitat, they actually had a higher natural germination percentage. So why did we have the low germination? First of all, we had an unknown seed characteristics. One of the exercises that should be done in every restoration project is to study the seed characteristics. What makes them not germinate? What makes them germinate? What are the limiting factors? Do they have some kind of dormancy or not? Do we need to treat them, and how? So once we actually study all those characteristics, we have a better understanding about how to increase and improve the germination rates of each species. And we don't need a fancy lab to do this. As in the case of Jordan, we just had very few resources, but were able to actually work with them to increase the germination rates. We had unknown limiting factors affecting germination post-sowing, and the seed trials performed in uncontrolled environments. maybe also we needed to focus more on controlling the environment for the seed trials, especially noticing that under natural conditions in forests, the germination was much higher. Maybe this was due to microsite that had preferable conditions, more shade, more humidity for the seeds to germinate, or just went under natural scarification. So all of this needed to be studied even more. So we didn't want to just improve seedling quality. We also wanted to monitor its long-term success on the site. We selected a point in the corridor, as you can see here in trial point 1 and trial point 2, between the Bossou area and the Nimba forest. We developed a small outplanting trial comparing new seedlings versus the old seedlings. And we worked with the local communities around the corridor to be able to make them own the project, and without the local communities, we can't have sustainability in restoration projects. So we worked a lot with not only the members of our partners, but also the local communities, as you can see here, in developing the trial on site. And you can see here how they are delineating on the left the plots, and then going to planting after selecting the places for the holes. The next step was to continue the seed germination research, to monitor the outplanting trial, and we're always in contact with them. And the results so far, the outplanting trial was so far good, and we always get report from on site. It's hard sometimes because it's very remote, but we're hoping that we can improve year after year the results, and develop a long-term restoration strategy by improving seed management, seedling quality, and outplanting practices. And here you can see that they also worked using local resources also on increasing native seedling production. So, which is what is really amazing about this is everything we use is based on local resources. In Morocco, we are working on a different story now about genetic variability and traceability. Morocco is such a diverse also country, and it has very high level of deforestation caused by livestock farming, drought, wildfires, logging, wood harvesting, as it's the same issue in many countries and most of the countries around the world. One of the main symbolic species in Morocco is the Citrus atlantica, the Atlas cedar. It's native to the Atlas mountain in Morocco, between the Rif, the Mid, and the High Atlas. It's listed in the IUCN as an endangered species and with a decreasing population, and it survives between the 1,300 to 2,600 meters. As you can see here, these are the Citrus atlantica seed zones. We have 3 in the upper atlas, 13 in the middle atlas, and 2 in the reef. So you can see even the ecological conditions, the seed zones ecological conditions are very different, from lower altitudes to higher altitudes, different climatic conditions, still for the same species. But we are noticing that we have a decrease in seed production in certain seed stands. And we need to ensure sustainability of the Citrus atlantica seed availability to continue restoration projects. So with this decrease in seed production and a poor seed quality, we also have an unknown genetic adaptation. For instance, how would a seedling from a certain stand in the mid-atlas adapt to another region or stand? So this unknown genetic adaptation has led this project in Morocco to focus more on genetic adaptation of this particular species. So just a quick review, what is a region of provenance? It's an area or a group of areas that is subject to uniform ecological conditions in which the stands, trees, they show similar phenotypic and genetic characters also. So here in Jordan, you can see that we have different regions of provenance depending on the climatic conditions and the climate type. So what we wanted to do in this project is to develop a certain genetic provenance trial. We wanted to study the potential genetic differences among different stands between the different regions of the Atlas seed zones in the Reef, Middle, and Upper Atlas, and develop this genetic provenance trial from seed collection to improved nursery practices to outplanting genetic provenance trial. So we wanted to understand the potential of the levels of genetic diversity within these stands and within this same species. So the consideration that we took is to identify the seed sources, to develop the nursery trial, and develop the outplanting trial that leads to analyzing the results about such genetic diversity. So this is, in summary, how we selected from 18 stands, we selected 8 stands, and in each stand, we identified 5 mother trees to be able to also improve our genetic provenance trial. So as you can see, if we take into consideration the mid-atlas, you can see them here in the blue tags. We have different climatic zones. So we wanted to make sure to group the ones that are geographically close together and select one that represents a cluster. So after doing this exercise with our local partners, we have identified 8 stands, and we focused on improving seed management techniques and improving nursery techniques from growing media to irrigation and fertilization management, keeping records. And this is now the current status of this trial. All seeds are labeled. We have a good germination, and we want to differentiate between the different families and different stands, seed zones, the different germination percentages. Also, it will tell us about the seed quality from each seed zone. So at the end, hopefully if we can travel again during this November, we will implement our outplanting trial, which from each of those 8 stands, we will be planting them in 3 different locations with different climatic conditions, one in the Reef, one in the Mid, and one in the Atlas, to see how they adapt and they genetically adapt to different conditions. All of this will lead to improve not only seedling production, but to monitor genetic provenance and to develop some kind of a traceability system with proper record keeping and monitoring. So at the end, we want to have the perfect seedling with being genetically adapted to a certain region and climatic conditions, knowing that that one species, the Citrus atlantica, it is so diverse genetically that it can adapt differently under different climatic conditions. Finally, in Lebanon, we have worked with the US Forest Service since 2011 and under different projects also. Lebanon has a very high flora biodiversity. The forests have important economic, social, cultural, and symbolic importance. However, we are, as many other countries, noticing decrease in the green cover, and its biodiversity is very affected. So all of our efforts was focused on developing research and developing a sustainability and restoration successes. So, and as you can see here, we have different ecosystems, from riparian, terrestrial forest, rangelands, and all of these ecosystems are affected and degraded in time. So we needed to focus on improving native plant material for restoration projects, taking into consideration the diversity of ecosystems that we have, and that every ecosystem has its own limiting factor. We started, I'm just gonna now go through a summary of some of the activities. We started with a lot of, we developed some mapping resources that were made public that shows where we did our restoration projects, and we developed vegetation maps in Lebanon, not developed but based it on other existing vegetation maps, fire risk maps, forest covers, and other resources that you can see on the website. And we started in 2011 with the most basic thing: seeds and seedlings. How can we improve seeds, seed management, and seedling production to improve our reforestation efforts on the long term? So we kind of we worked a lot and collaborated a lot with national and international academic institutions around the world, and we collaborated with universities in Lebanon and outside of Lebanon, in the US, Spain, and Turkey, to be able to develop more precise seed germination protocols based on scientific approaches and lab testing. We also didn't want to use very sophisticated resources, so we kind of stick with the idea to use what is available locally. And here you can see a nursery manager, Farouk, that is using a slide to do the seed separation by weight. A lot of other good able to separate seeds and clean seeds. So since 2011 until 2019 and 2018, we're able to work a lot on nursery management practices, starting from seed to seedling production. And we were able to improve native seedling production and developed a national guideline for nursery management. And this also focuses a lot on seeds. This guideline focuses a lot on seeds and how to improve seed management practices for each native species. And we have around 40 native species in that guideline. Other guidelines that because of all the trials that we did and the research on the ground, and it's practical research, not necessarily peer-reviewed articles, but a lot of it is based on practical research on the ground. We developed some outplanting and reforestation guidelines. We worked a lot on mapping resources, fire, climate change, and we were able to see a lot of success in planting more than 1 million seedlings, with survival averages ranging between 50 to 90% and plus throughout the country, depending again on the climatic conditions and on the limiting factors on the site. And this is also thanks to the local communities that were involved in each of the projects. Something that is new, and I'm going to end this presentation with this idea, is the role of fauna and ecosystem. And here I need to thank my colleague Samara, a zoologist in Lebanon. She has taught me a lot about this, and we are currently working on this issue more as research in LRI in Lebanon, and what is the role of fauna in improving ecological restoration. So we all know that the role of fauna in an ecosystem, they have an important role as consumers and decomposers. Animals are very important component of an ecosystem. They maintain a certain cycle of energy in the environment, and they ensure sustainability of habitat. So even if we come there and we introduce native species and we follow and we monitor and we take care of our restoration projects, but we need to think on the long term, how can this ecosystem be sustainable and functioning again without us interfering the whole time? And this is where the research in fauna and ecosystem is very important. The fauna-flora interaction creates a certain ecological balance. And we have so far just based the success of restoration projects based on just the abiotic and the flora-based criteria, such as successful of the vegetation survivor rates and etc. And we have not monitored fauna enough, and we have neglected in its role in ecological restoration assessments. So fauna being overlooked and understudied and the process of recovering functioning ecosystem has pushed us to look further and deeper into this issue. So we are starting, me and my colleague, we are starting to work on different techniques to assess fauna and restoration using non-invasive techniques. So I'm going to talk about 3 non-invasive techniques in this presentation. One that is environmental DNA, eDNA, footprint identification technique, and DNA analysis through sprint collection. What's environmental DNA or eDNA? eDNA describes the genetic material of organisms present in the environment. So if we are in a certain ecosystem and we're not sure what is living in this ecosystem, eDNA will help us in identifying the species that live in this ecosystem. It can be applied to a large range of ecosystems from aquatic to terrestrial. And the eDNA metabarcoding, it can characterize multiple the DNA of multiple species in one single sample. So it's the application of eDNA metabarcoding include biodiversity monitoring and more. So this is just a very quick summary of what are the applications of eDNA metabarcoding. And now I will tell you in the next slide how we use it and how we actually can sample for it. It can identify ancient ecosystems. It can tell us the plant-pollinator interaction, which is very important in natural regeneration of a certain ecosystem after restoration has been implemented. It can tell us the diet analysis of a certain species. What does this certain species feed on? Which plant or which other animal? And it tells us what plant is important for them to feed upon. And it can tell us if we have some invasive species that we cannot visually detect, but through eDNA we can detect. It can also be applied to as a pollution response and air quality monitoring. So it's a very simple technique, and we're gonna start our first sampling hopefully at the end of the month, where we're gonna collaborate with a lab in the UK, Nature Metrics. We basically select samples from the river or nearby water body. We collect the sample, and we perform a sequence of PCR, qPCR, metabarcoding. Several species are identified, and as you can see here in this table, it identifies all the species and the DNA that is present from that one sample, and it can also tell us the density of the population at the time we have collected the sample. Another technique, non-invasive technique, is the footprint identification technique, or FIT, that was developed by this organization called WildTrack. And again, we are starting this collaboration with them. So basically, as the name says, you use a footprint to identify species, individuals, the age class, and the sex. It's a non-invasive technique that can work with other non-invasive techniques such as eDNA that we mentioned before. It is very cost effective, and it can obtain high accuracy. And it's accessible to traditional trackers. Anyone can go on the field. All you need are rulers, a smartphone camera, and this EpiCollect app that you can download on any smartphone. And we start tracking footprints, take pictures of the footprints, and it can identify the species, the individuals, their age, their distribution. So it's another non-invasive technique that helps us monitor fauna and ecosystems. One last technique that I want to mention is the sprain collection, which is basically analyzing DNA from collecting sprain. The author in Lebanon, Lutra, is it was mentioned that it was critically endangered in Lebanon. So we decided to work on two zones where we know that they have been seen there. So by collecting their sprains, we can determine their diet trends, estimate their population density, the genetic structure of that population, and estimate the genetic variability and spatial organization. Simply collecting the sprains, analyzing the DNA, and it can tell us all about the diet trends, density, and genetic structure of the population and its genetic variability. So I'm going to end with this slide. Why is fauna assessment very important in ecological restoration? It actually serves us as a baseline for fauna and flora interaction in natural habitat or restored sites. It will help us guide in selecting the right native species in restoration projects, depending on which fauna and native fauna we want to be promoting also. And it could help us in achieving a better understanding in the process of recovering functioning ecosystems as a whole, not just based on flora, but based on this whole fauna and flora interaction. So I'm gonna end my talk here and just sharing my email with you here. And a big thank you for the implementers and partners that we have worked with in all those amazing projects and countries. And I guess this is it for now.
Thank you, Karma. We already have quite a few questions in the questions box, so please feel free to add them in as we're going, as you think of things you'd like to hear more about. So the first question is, it has been suggested that seed germination in a native soil encourages plant colonization by native microbes. Have you used or investigated the source of microbes in your restoration projects?
No, we still didn't. And this is something that we need to be looking more into details also in our projects.
Great. And there's also quite a few questions about eDNA. So the first one about that is eDNA used very much for flora or primarily fauna?
So this is a new technique that we are actually investigating now, and it's used more for fauna. So from amphibians, invertebrates, vertebrates, mammals, it can be used for a large number of species and families. But it's very important because it can tell us what species are thriving in this ecosystem, and it could help us in guiding our selection of native species. So I will answer now based on the knowledge I have about eDNA so far, and if you can ask this question 2 months later, we would have our first results. But for now, it's specifically being used for fauna.
Okay. And several people are curious if you can use it in, is eDNA primarily used in aquatic environments? If it's used in non-aquatic environments, what are you collecting?
So it could be used in it could be used to determine your fauna and species in aquatic and terrestrial terrestrial. So how this works, and we were part of this webinar that they gave, which was very interesting. And they told us that the best time to collect for terrestrial ecosystems from rivers or from other water bodies is after the rainy season, because with the rain and the melt of the snow, or the rain after the end of the rainy season, all the DNA that was collected from higher elevations was drained down to the river. And that when we collect that sample from the river, for example, it's not only representative of the fauna living just around the river, it's representative of the DNA from the fauna living also on the terrestrial parts. I don't know if this is making sense a bit or not. So when we were in contact with that lab, they told us the best time is actually now at the end of your rainy season, which is now. So we're hoping that next week we receive the samples and we can go and collect some samples. And we will be gladly happy to share by email the results to anyone who's interested from this webinar.
Great. Thank you. The next one, also about eDNA, is what capacity is needed to do eDNA? Can this type of analysis be done in Guinea, for example, without sending the sample to another country?
It depends on the resources and the lab resources that they have. Very few labs can do the eDNA meta barcoding, and we are trying to ship the samples. They have worked with a lot of countries in Africa and Europe and other continents to ship the samples to their labs, which tends to be easy and not expensive, because by weight it's just a small tube and it weighs very little also.
Switching topics, the next question is, how have the fires in Lebanon and predictions for reduced precipitation due to climate change affected your restoration projects?
So this is something very interesting, because now we're having a heat wave in Lebanon, and the fire risk map is all red in
No.
Lebanon. So it's actually helping us in managing better and being more prepared in fire prevention. And so developing models to know that when it is a high risk for fire season in Lebanon has helped us a lot to work with the local communities to be able to prevent fires. Last year we had huge fires all around of Lebanon, and it was very unfortunate, and it was very quick. And so we try to work hard to integrate all the data that we have to be able to manage our forest and our restoration sites. But so far, data has been helpful a lot in preventing a lot of the fires. So I hope I understood the question right.
Great. The next question is, has your collaboration with the local women continued? And if so, what is their current role?
Yes, and it's continuing, and we are actually now starting to learn from them because they are working in the nursery every day, and we are in constant contact with them, not only from nursery visits, but to giving them trainings, continuous trainings about different topics. And they have kind of emerged as leaders in that field. They have taken that project, and it has not only empowered them, but it gave them some kind of an ownership to that project. So working with the local communities and giving them the technical resources to be able to implement a project is very important. in every project that we have worked on, when we support the local community in any technical capacity building, and not only giving them guidelines on how to do a certain project, we have found that that local community is working with us on the long term and supporting us in restoration projects.
So, do you have any suggestions or tips on how to ensure and maintain restoration efforts beyond project funding ending?
Something that me and my colleague were talking again about is the data. Data is very important, and this is why we're looking at some techniques with high accuracy for data collection, like eDNA or other kind of data, because our limiting factor in having proper restoration projects and ecological restoration is the lack of accurate data that we might have. So we are kind of focusing now in our research and development program on trying to identify the data gaps and how we can actually fill those gaps to be able to improve restoration practices. The more data we have, the more accurate data we have, the more improved the restoration projects can be, and their cost can even decrease more. So we can all just focus on really generating a lot of data.
Mm-hmm.
And one set of data in one country could be helpful for another country also. So it's all interlinked.
So we had another question about eDNA, and
Yes.
it was just to try to get some more information about how what you collect is used, for example, to find evidence of plant pollinator interactions.
Mm-hmm. So we still didn't try it, but what we have learned from this webinar is we can identify the pollinators that are present in a certain area, and from this to see which native species are important for those pollinators to thrive also, and from which species are important for those pollinators to survive. So this is how, and again, we would be more than happy to share the results of our first trial with eDNA for this. Hopefully it will be during this summer. We were promised to get our first kits hopefully next week and ship them back the week after, and we can get our results. And we would be more than happy to discuss more about the eDNA findings and how it would help us in our restoration projects, because this is still a new idea for us. But focusing on the pollinators and the presence of wild pollinators is really important in our projects and for the sustainability of restoration projects and the functionality of ecosystems.
Great. And for this question, I think the person who asked it is probably thinking about a specific project where you were collecting seeds from remnant forests. And so the question is, do you only collect seeds from remnant forests, or are there other sources for seeds when you are trying to collect from less common species?
Okay, so basically in most of our projects, we focus on identifying seed zones which are existing forests. So we don't have specific seed orchards or areas that were developed and just for seed collection, but we kind of focus on identifying the best forest stands for seed collection. So there are already existing forests, but after several years of collecting from those forests and identifying their seed quality, we can identify as a suitable seed source or not. But we mainly focus on existing forests to identify seed sources, but not on other, such as seed orchards or.
Karma, I think did you cut out or did you?
Oh, I don't know, maybe.
Oh, okay. All right. I think we have time probably for 1 to 2 more questions. The next
Okay.
one is, in relation to climate change, many restoration projects are moving genetic stock to preempt changes in the environment, like moving seedlings from heat-tolerant populations to areas with predicted increases in extreme heat events. Is this
Mhm.
something you are considering doing in your projects?
But I think I'm gonna take the example of Jordan. Can you just repeat the question a bit? Sorry.
Yes, no problem. So in relation to climate change, many restoration projects are moving genetic stock to preempt changes in the environment. Things like moving seedlings from heat-tolerant populations to areas with predicted increases in extreme
Mhm.
heat events. Is this something you are considering doing in your projects, trying to match your seed sources to future climate?
Yeah. So again, I will give the example that we did in Morocco, where we're trying to find the best to develop a traceability system where we know that seeds from a certain stand would be genetically adapted to certain climatic conditions. So it's two things we can focus on. It's either the actual condition now, or if we can do modeling. For instance, we use a lot of modeling to kind of predict how in 10, 20, or 50 years climate change would affect the actual climatic conditions in a site. So, but for now, we focus on the actual climate at present, and we use genetically the plant material that are genetically adapted to those conditions. But doing this could be good in a lot of areas where modeling is predicting a shift in climatic conditions and a shift in species distribution also. So again, ecological restoration is so broad, and there's a lot of factors to consider. But when we work on those projects, we try to identify the most limiting factors we have and start here as a baseline and as a start, and then build on this.
Thank you so much, Karma. Could you put the slide up with your website on it one more time?
Yes, if I can. Oh,
Well, thank you everyone for
okay. This
participating
is
today. And thank you especially to Karma for this wonderful presentation. If we didn't get to your question, which I know there was quite a few we didn't have time to get to, you can follow up directly with Karma. We're also planning a webinar in July on eDNA, so if you're interested in that, please keep an eye on our website. You'll be getting a follow-up email with the certificate of completion for today if you need one, as well as a survey to give us feedback on today's event. As I said at the beginning, we have recorded this webinar and it will be available on the SCR website. I encourage you to visit our website not only to find that recording if you want to share it with anyone, but to learn more about our work and the benefits of membership with the Society, including access to our entire webinar and conference library. Please be safe and healthy, and we hope you'll join us again next time.